Neural communication requires movement of ions through the neuron cell membrane. Neural signals result from electrochemical changes. So remember how Helmholtz, Galvani, and others determined that the nervous system used electrical signals? It is chemical changes that lead to these electrical impulses. The cell membrane is very important in neural communication. So let's take a look at how it works and what it's made out of. It's made out of lipids, fats, right? So these fat molecules or lipid molecules are arranged in such a way that they form what's called a semi-permeable membrane around the cell. And the way that works is that the heads of these lipid molecules are attracted to water, and so there's water in and out of the cell, and so the head area will be the one that's in contact with the edge, either the inner edge or the outer edge. And then in the center, the tails will come together. So we can see here that there's on the outside of the neuron, there's a layer of where the heads are, and on the inside of the neuron, there's a layer where the heads are, and then in the middle here, kind of like a forming two bread slices, sort of, is the middle part, and those are the tails of these lipid molecules. And proteins hold the cells together, and they control the environment in and around the cell. So we're going to see that fats are very important in your brain's health, as are proteins. And your book has a little section towards the end of the chapter where it talks about fish oil, so omega-6 fish oil being really important and beneficial in a lot of ways. And this is just another way that they're very helpful, is with keeping these cell membranes functioning properly. So the neuron has what's called a selectively permeable membrane. Water and gases can pass freely through. Right, so those can just flow through as needed. Other substances, however, are barred from entry. They're not able to pass through. And so, for example, like a very large molecule might not be able to go through at all. Others can pass through protein channels in the cell membrane. So here we see this sort of what looks like kind of like a gate. That's the protein channel that will allow certain substances to flow in and out of the neuron as needed. We see that there's a polarity or a polarization between the inside and the outside of the neuron. So it's the difference in electrical charge between the inside and outside of the cell. And that polarization is going to be important as well. The resting potential is the difference in charge between the inside and the outside of the membrane when the neuron is at rest. So you can think of the neuron as sort of like your heart. And your heart beats, but there are also periods when it's not beating, right? So it'll beat and then take a little break, beat, take a break. Neurons are kind of like that, although their activity is not necessarily quite as rhythmic, but they are going to be having what are called action potentials, which we'll learn about in a little bit. So that's sort of analogous to like a heartbeat. And in between, though, they are considered at rest. And so that's the resting potential, is sort of them just sort of hanging out, not really doing anything. It's sort of their default position. This default position, this resting potential, is caused by the distribution of positive and negative ions. And it's the distribution of the ions inside compared to the outside of the neuron. And we see that in different neurons it'll range, but it's always negative. So it'll be either between -40 and -80 millivolts will be the resting potential in different neurons. In this class, we will usually just use a standard -70 millivolts. So that's what we'll go with, sort of the typical resting potential being about -70 millivolts. Now, what ions are found inside and outside of the neuron? Inside, there's mostly potassium, and you can see here that we're going to be using the symbols for these different ions. So potassium is K, And notice it has a little plus. That's telling us that potassium is a positively charged ion. There are also these large negatively charged protein ions, which get an A minus. The A is for an ion, which is a chemistry term for a negatively charged ion. And we see, of course, the minus because it is negative. Less common inside the neuron are sodium, Na+, so sodium has a positive charge, and chloride, Cl-. And if sodium chloride rings a bell, it should. That's because that's table salt, right? And so anyway, so there's basically some salt inside the neuron, but not very much. Outside the neuron, it's mainly composed of sodium and chloride and a smaller amount of potassium. And so we can see here in this picture, the sodium ions are depicted in red. The and the chloride ions, I believe, are depicted in green. And then the potassium ions are depicted here in blue. So you can see that there's a greater concentration of sodium and chloride on the outside and a greater concentration of potassium on the inside. Now, how do these ions move across the cell membrane? There are three basic ways that they can move. They can diffuse, and that's really just a process where ions flow from where there's a lot to where there's less. So diffusion has to do with the quantity, the number of these ions. So they will naturally flow from an area that has a high concentration to an area that has a lower concentration. And if you remember being a kid at some point in a basic science class, or just noticing this observation that if you put a spoonful of sugar or salt or something like that in a glass and you don't stir it, you just leave it alone. If you wait a little bit, the salt or the sugar, instead of being accumulated at the very bottom of the glass, will eventually diffuse, right? It'll distribute itself and kind of naturally mix in the water. So that's diffusion. Electrostatic pressure has to do with the positive and negative charges on these ions, right? So if we remember, all of these ions, sodium, potassium, chloride, have either a positive or a negative charge. And this is important because in the world of electrostatic pressure or electrostatic charge, opposites attract. So positive ions want to go towards negative ions and vice versa. The opposite is also true though. Same signs repel from each other. They would want to get away from each other. So two ions that both have a positive charge are going to want to go away from each other. And this also applies in this case to if there's a positive ion on the outside of the neuron, like this red sodium, and the inside of the neuron has more negative ions, then guess what? Every positive ion out here, including the sodium, wants to go in. It's going to be pulled in, if possible, due to electrostatic pressure. So that's another way that opposites attract. The positively charged sodium will want to enter into the negative area inside of the neuron at rest, because remember, the resting potential of a neuron is about negative 70 millivolts, right? The third way that ions can move in and out of the neuron is by using the sodium-potassium pump. The sodium-potassium pump uses energy, so it's not free. These other two, diffusion and electrostatic pressure, they're free. They just use physics. The sodium-potassium pump is not free. It requires energy. And so what it does is it moves 3 sodium ions out for every 2 potassiums that it moves in. So it's exchanging sodium for potassium. And this allows it to keep the neuron negative. Why? Because it's always kicking out an extra positive ion. So 3 positively charged sodium ions get moved out for every 2 potassium ions that get brought in. So the more that this happens, the more of the sodium ions are outside, and that means there are fewer positive ions on the inside.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.